The overall goal of this procedure is to use an impedance-based technique to follow the real-time cytoprotection of a neuronal cell line to delineate the involvement of second messenger pathways in the protection process. This is accomplished by first culturing a neuronal cell line under proliferative conditions in an impedance-based real-time cell analyzer. In the second step, potentially relevant second messenger pathways are pharmacologically inhibited before subjecting the cell line to neuroprotective treatments. Next, the status of the neuronal cell line is followed using a real-time cell analyzer under the different experimental conditions. Ultimately, statistical analysis can be used to assess the real-time involvement of second messenger pathways in the cytoprotection of the neuronal cells.
The main advantage of this technique over existing methods, like endpoint neurotoxicity assays, is that it allows cytoprotection and cytotoxicity to be evaluated in neuronal cell lines in real time and under label-free conditions.
This method can help answer quick questions in the molecular neurobiology field by contributing to more high-throughput assessment of potential neuroprotective compounds in vitro, and by providing insight into the molecular mechanisms involved in neuroprotection.
Begin by washing an adherent neuronal cell line layer. For example, here, human neuroblastoma SKNSH cells are used with PBS. Then trypsinize the cells for 5 minutes at 37 degrees Celsius.